Good morning. So I'll be talking about autoimmunity and immune tolerance. So basically, let's get a summary of what it is. The immune system must discriminate between self and non-self, and when it fails, it is termed as autoimmunity. This is achieved through mechanism of immunological tolerance, which suppress or eliminates the self-reactive lymphocytes. And when the tolerance fails, the immune system can inappropriately target the self-antigens, resulting into autoimmunity. The autoimmunity diseases are often chronic, inflammatory, and multifactorial, involving genetic predisposition, environmental triggers, and breakdown of self-tolerance. Also, understanding the balance between tolerance and autoimmunity is central to developing therapies for disease such as type 1 diabetes, rheumatoid arthritis, and multiple sclerosis. So coming to the mechanism of tolerance, there are two types: central tolerance and peripheral tolerance. The central tolerance is the first checkpoint in the prevention of autoimmunity. It occurs during the early lymphocyte development in primary lymphoid organs, and its goal is to eliminate or inactivate the self-reactive clones before they enter the circulation. So for T cells, there are two types. It is positive selection and negative selection. The positive selection occurs when the in the cortex of the thymus, where thymocytes expressing the T cell receptors must be able to recognize the self-MHC molecules presenting self-peptides. So if they cannot interact with the self-MHC, they die by apoptosis. And negative selection where occurs in the medulla of the thymus, where the thymocytes whose TCR bind to self-antigen MHC complexes with high affinity will undergo apoptosis. This is mediated by medullary thymic epithelial cells, which expresses tissue-specific antigens via the transcription factor autoimmune regulator, enabling the self-reactive T cell deletion. And this process also removes the potentially autoreactive T cells that could in future cause autoimmune diseases. So only the T cells with moderate affinity for the self-MHC complexes survive and become single positive CD4 plus cells or CD8 plus T cells. So some self-reactive T cells may differentiate into regulatory T cells or for the peripheral suppression. For the B cells, they undergo the three steps, that is clonal detection. If immature B cells are strongly bind with multivariant self-antigen, they undergo apoptosis, or receptor editing, where if the B cell recognizes self-antigen with high affinity, they might rearrange into light chain genes again and produce a new non-self-reactive BCR. And energy, it is the B cell will recognize soluble or monovalent self-antigens, becoming functionally inactivated and unresponsive in term that is termed as energic B cells. So these cells may enter the periphery but are short-lived and non-functional. Coming to the peripheral tolerance, the peripheral tolerance functions as a backup system to control the self-reactive lymphocytes that escapes the central tolerance. So it is occurs in the secondary lymphoid organs and in the peripheral tissues, and is critical for maintaining the immune homeostasis, especially in non-thymogenic agents or antigens not presented during the development. So for the T cells, the following steps undergo for this process. The T cells will require signaling for activation. So the first signals comes by the TCR recognition of antigen MHC. The second signal will come by co-stimulation of via CD28D7 interaction. So if the signal 1 occurs without co-stimulation, the T cells become energetic, unable to respond even upon the future encounters. The suppression by regulation of T cells, the natural traits arise from thymic selection, while the induced traits differentiate in the periphery. So they express the CD4 plus, CD25 plus and FOXP3. The suppressed responses, it is by cytokines, interleukin 10, TGF beta, or by inhibitory receptors by CTLA4 or PD1. The modulation of dendritic cells and other APCs are done by this process. So the activation induced cell death. It is the repeated or chronic stimulation that will lead to expression of death receptors, especially for FAST, the CD95. In the interaction with FAST ligand triggers apoptosis and self-reactive T cells. It is important in shutting down the T cell response after infection or inactivation. The immune privileges. Certain tissues such as eyes, brain and testes express low level of MHC and produce immunosuppressive factors, limiting the immune response. Physical barriers and absence of lymphatic drainage can also contribute to limit the immune response. For B cells, the process of energy is same. So the B cell will encounter a soluble self-antigen in the periphery. Without the T cell help, become unresponsive and eventually will die. The lack of T cell helps. Even if the B cell will not bind to the self-antigen, they require T cell derived cytokines and CD40, CD40L interactions to become fully activated. So without this, the B cell will always remain inactive. And in process of deletion, strong self-reactive BCR signaling can also induce apoptosis even in the peripheral B cells. Coming to the mechanism of autoimmunity, there are certain triggering factors which will cause autoimmunity. They are mainly environmental or intrinsic immune defects or genetic factors. So the autoimmunity, it is a result of failure of self-tolerance. It will leading to activation of autoreactive B or T cells. So it may be organ-specific and systemic. So the organ-specific or systemic, we will discuss this later. And this breakdown is often multifactorial. So the factors responsible are environmental triggers, such as infections that can trigger autoimmunity through molecular mimicry, that is when pathogens antigen resemble self in case of rheumatic fever or streptococcus infection. Bystanders activation, it is infection leading to general inflammation activating the autoreactive cells. Also epitope spreading, it is the initial immune response broadens due to the self antigen broadens to include the self-antigen. The intrinsic immune defects, defective traits or abnormal antigen presentation can promote autoimmunity. The aberrant cytokine production, for example, like excessive interleukin 17 or INF gamma, will derive inflammation tissues and cause damage. The genetic factors, certain factors such as MHC alleles are strongly associated with autoimmune risk. For example, HLA-B27 or HLA-DR4. So these mutations in FOXP3 or CTLA4 can disrupt the tolerance. There are certain other mechanisms such as neoantigen formation. This is when the tissue injury or drug modification will create a novel self-antigen. Also, in cases of loss of immune privilege, trauma or inflammation exposes the sequestrated antigens. For example, in cases of symphatic ophthalmia. So the autoimmune disease, as I said before, it it can be organ specific or it can be systemic. So organ specific examples are type 1 diabetes, where the T cells destroy the pancreatic beta cells. In Graves' disease, the autoimmune the autoantibodies stimulate the TSH receptors, and in myasthenia gravis, the autoantibodies will block the acetylcholine receptors. In systemic, there is systemic lupus erythematosus. So in this, the autoantibody is created against the nuclear antigen, form immune complexes. Rheumatoid arthritis, the inflammatory T cells or B cells responses in joint system, where the presence of rheumatoid factor is present against the IgM against the IgG. And in cases of multiple sclerosis, the T cells will target the myelin sheath in the CNS. There are certain more examples such as psoriasis that will occur in the skin, or such as some neurological disease, and in GIT some disease can occur such as ulcerative colitis or Crohn's disease. So coming to some animal models that are studied are used to find out the autoimmune diseases. So experimental models, the animal model will have greatly enhanced our understanding of autoimmunity. For example, in cases of non-obese diabetic mice, the model for type 1 diabetes, it develops spontaneous B cell destruction. Also, experimental autoimmune encephalomyelitis, it is the T cell mediated demyelination mimicking multiple sclerosis. In collagen induced arthritis, it mimics the human rheumatoid arthritis. It shows the same symptoms. Then iron knockout mice, it is used to study the multi-organ autoimmunity due to failed central tolerance. There are some recent research that have undergone in this area. So such as the CAR tracks for autoimmune. So it is engineered regulatory T cells that are being explored to suppress the specific autoimmune reactions while preserving the general immunity. There is neoantigen profiling in autoimmunity. It is the advanced proteomics now allows for the identification of self-modified self-proteins driving diseases like rheumatoid arthritis and systemic lupus erythematosus. The microbiome and autoimmunity. So dysbiosis has been linked to the lupus and MS. So commensals may educate the immune system to tolerate or self-reject. Also, the checkpoint dysregulation in autoimmunity. The PD-1 CTLA4 mutations have been implicated in the systemic autoimmunity, and the therapies blocking these checkpoints it is used in cancer, so it can induce the autoimmune like adverse side effects. So that is all for the autoimmunity and immune tolerance. Thank you.